Okay, good morning, Bio1 students. Just so we're clear for the last quiz, which will be quiz 8 for you, it's going to cover 25 definitions from 26, 27, which is bacteria, and 28, right? 28, which is protists. So, as I was saying, if you take a look at the note outline from 25, at the very end of it, there are definitions, the words you need to know for 26. So those will show up on the exam, either as matching just all of them together. I think there's 10 of them. 1, 2, 3, 4, 7, 8, 9. There are 10. So either the 10 of them with match the terms, or if I put them as multiple choice, they'll be which of the following definitions is mismatched with its term. So expect that. And this is my mocked up version of the exam. So it looks like it's done. So that's good. And it looks like it's pretty evenly balanced in case you're interested. It looks like there are 10 questions from 25, 10 from 26, 10 from 27, and 10 from 28. So it looks like pretty even Steven. And it looks like it's going to be 1, 2, 3, 4. Why can't I do this? So it looks like it's going to be about a 40-point final exam. So today, since I just checked on Canvas and there is a set of PowerPoints for 25, I would focus on those PowerPoints to study. But there isn't a video or PowerPoints for 27. I thought I would like to go over 27 with you. So let's start with that today. And I'm probably going to end up using my ye old whiteboard because I am on the laptop, not the tablet, so I won't be able to draw unless I draw on the board. So there. So chapter 27 is such as it goes, like 16 and 17, one of my favorite chapters in the book. And the reason for that is because it's about bacteria, which is probably something that's probably what I know the most about. That and some little bit of immunology, a little bit about viruses. So let's take a quick look at 27. There is an extensive note outline for 27 that's up there. It's labeled summer 2020. So that's good because it means I just sort of updated it for this class. So let's start at the beginning. I'm going to move over to the board here. Try to draw a little bit instead. So I'm going to get rid of all of you. Make the gallery small. Just click you like that. Okay, there. I can find a marker that works here. All right, so bacteria, remember, they belong to this domain, the domain archaea. Right, and we've got 2 kingdoms. So we've got 2 domains actually of bacteria. These, the extreme, I like to call them the extreme bacteria or prokaryotes. And the domain, just bacteria, we call it now, right? And they've got about 5 kingdoms divided up into this. Now, we used to argue about that quite a bit, but it looks like we're settling on 5. So 5 kingdoms in domain bacteria. And then of course, the third domain, I'm trying to keep you fresh and remind you for the final, right, is eukarya. And that's going to have the kingdoms of eukaryotic cells, which would be the protists, which we'll talk about on Monday, the fungus, the plants, and the animals. Right, so that has 4 big kingdoms under it, although there's a lot of debate about making more protist kingdoms. Right? So today we're going to focus on these guys, the domain bacteria and the domain archaea. And number one thing I would remind you that I would, if I were you, I would go back to chapter 7. Chapter 7, tour of the cell. And I would make a new list of comparing prokaryotes with eukaryotes. So today, since we're talking and focusing on the prokaryotes, since both of these domains are prokaryotic, I'm going to just make a quick list or just a rundown of the big highlights of the prokaryotes. And I would say to you, first and foremost, they lack a nucleus, right? We say lack a membrane-bound nucleus. They lack membrane-bound organelles. Yeah, didn't spell that right. Remember, their DNA is circular. They have one chromosome only. And it is circular. So they can replicate much faster. They do not have mitosis or meiosis. They reproduce asexually by a specific mechanism that we call binary fission. Their DNA does not have histone proteins, so it's not wrapped around proteins to make it compressed or fit smaller inside the cell. They are usually about 10 times smaller than eukaryotic cells. Usually put them in the range of 1 micrometer in diameter to 10 micrometers. That would be a giant bacteria. Eukaryotes 10 times larger than that. A red blood cell is 10 micrometers. And if you tried to look at a red blood cell, well, you did. You looked at red frog blood under the microscope. Oh, no, you did not. Sorry. If you looked at frog blood or human blood under a microscope, some of the smallest cells we have are red blood cells, and they are about 100 micrometers in diameter, up to eukaryotic cells about 100 micrometers in diameter. I'm not going to fill out all the contrasting eukaryotic things. You guys can do that on your own. Okay. So this is always where I start this chapter, because it's really important that we sort of bring it all back to the beginning of the semester. And it's a good time for us to think about, just like all the thousands of things you've learned this semester, and how much more sense this list makes to you now than it did when we made it at the beginning of chapter 7, when we started to tour the cell. Or 6, actually. It's chapter 6, isn't it? Come on, you guys, catch me on those things, right? So that chapter 6, towards 7 was membranes, right? So now we're going to shift our focus entirely to just talking about these domains. And I'm going to primarily focus on the this domain here. Oh, wait, I have 8 colors. All right, so I'm going to mostly focus on the domain bacteria, not archaea. We'll just talk a little bit about archaea. Want to know more about them? Come to microbiology. Learn all about it. Sign up for my classes so that I still have a job in the spring. And you do get a lab kit in microbiology that does come with a microscope, so that's very exciting. And I heard tell that second round of CARES money, as they'll be able to use more of it to help students with purchasing the lab kits. I know that's not something you guys want to hear. It's good because more people will actually buy them. A little taller, so have a little better focus. All right, so let's talk first about the shapes. So for bacteria, we call it, particularly we say kind of old-fashioned, we call it morphology. And that means the shape. And they come basically in 3 shapes. We have a sphere shape, which we call coccus, a rod shape, which we refer to as bacillus, and then there's a category that is called spirals or curved. And we have a couple of names for those depending on how spiral or curved they are. So I'm going to list those out separately underneath here. So there is literally one that's curved and kind of looks like a comma, like this. It kind of goes like that. Those are called vibrios, vibrio shape. There are soft spirals, or almost like a squiggle, that we call spirilla. And they look like this. They almost look like a piece of pasta if you threw it down on a counter and it kind of just made a wavy shape. And then there are also ones that we call spirochetes. And spirochetes are actually corkscrew shaped like that, but in 3 dimensions. But if you look at them under the microscope, they just kind of look like a zigzag like this, because you can't actually see the curve unless you have an electron microscope. Sphere shape, obviously, like this. Bacillus, or the rod shape, they can be small like that. They can look almost like a coccus. We call those cocobacillus. Or they can be kind of long and sometimes almost clubbed. So sometimes they're in chains like that. So when you see the words, yes, that's it. We just left it like that. These can occur in groups, clusters. If they occur in clusters, this is not something for the test, but just words that you may have heard. If it occurs in a cluster, we have a name for that. We call it staphylococcus. And if it's a chain, like I drew here, that's a streptococcus. Well, this would be streptobacillus. Let me try again here. All right, so that's that as far as morphology. One of the most important features of bacteria is its cell wall, which exists outside of the cell membrane, of course, right? Got the best angle on that, so we get the most coverage. There we go. You'll see it abbreviated like that, CW for cell wall. The cell wall of bacteria has a unique chemical in it. It's called peptidoglycan. Because it has, these are words that you might know now, right? Because it has peptides in it. So a short chain of amino acids. Ooh, chapter 5, never lose it. And glycan, see the word glucose in there, because they're modified glucose molecules. This peptidoglycan only occurs in bacteria, only in prokaryotes, never in eukaryotes. So unlike something like chitin, which shows up in fungus and in animals, or something like cellulose, which shows up in plants, only this is a bacteria only, the peptidoglycan. And why it's important is because depending on the quantity of this and its placement, so I'm just going to say that, quantity and location, it divides pretty much all the bacteria up into 2 large groups, which we call gram-positive bacteria and gram-negative bacteria. And again, I'm not going to bore you with super details about this. What is important to remember is that because of the quantity and location of the peptidoglycan, we've got these 2 different groups of bacteria, which we determine by staining them and looking at them under the microscope. The stain is called the Gram stain, and it's named for the man, that's why it's capital G, Hans Christian Gram, in the 1880s, who developed this stain so we could tell the difference between these 2 groups of organisms. And again, the take-home message is you treat them with different antibiotics. So this is the first thing that is done if you have an infection. It's suspected to be a bacterial infection. And then based on this result, they're going to start an antibiotic specific to whether it's a gram-positive or gram-negative cell. They're not going to be able to tell the name of the cell that first hour, but they can do this test and immediately start drug before the organism has a chance to even multiply even more. All right, some other interesting features of bacteria, other interesting structures they have that help with what we call their virulence, or making them really good at making you sick, let's say. Those would include things like some bacteria have a capsule. This is a protective coat outside. So here's phospholipid bilayer, right? The cell membrane. Here would be the cell wall. And then some bacteria outside of that have a third coating. That's the capsule. Capsules are very common in respiratory bacteria. Bacteria that cause respiratory infections, let's say, respiratory pathogens. Pathogen just means a bacteria that can cause a disease because they're very protective having a capsule, as you might imagine. The capsule is sticky, so it allows bacteria actually to form groups, and we call them biofilms, almost like little rainforest ecosystems. You want to think of it like on your teeth. First bacteria stick that have a capsule, and then another bacteria can stick to them, another bacteria can stick to them, and they can make this whole community that interacts and is better at making you ill than if it was just one of them. They also help protect the bacteria from drying out. So that's a good thing if you're a respiratory pathogen, right? If I sneeze on something and let's say I have a streptococcus infection, streptococcus pneumoniae infection, and I sneeze on some sort of like a hard surface, and then I walk away, the bacteria on that surface can survive longer if they have a capsule because they don't dehydrate. Someone else comes along, touches the computer, touches the mouse pad, has an itch, puts it in their eye, up their nose, their mouth. For some reason, they have the potential to then get that infection. All right, so fimbriae. They show some pictures of fimbriae in your book. These are super interesting structures. They're extensions that come out of the bacteria. So here's again cell membrane, phospholipid bilayer, and then cell wall outside of that. And then if the bacteria has fimbriae, they stick out like this all around. Sometimes they're really long, sometimes they're kind of short, right, like that. Those are fimbriae. They are for attaching, or we say adherence. And it's just been shown that they can actually help the bacteria move. So they can actually be involved in motility, which is a new thing, new feature of fimbriae. They can also be used, we'll talk about in a minute, for exchanging DNA from between one cell and another. Some bacteria also have flagella. We can go on and on. The flagella are different than the fimbriae in that, so here's the bacterial cell, here's the cell wall. The flagella have a particular attachment, it's complicated, and then they tend to be longer, but they can in singly, or sometimes the bacteria might have a whole group of flagella at one end, like this. Almost we call it like a tuft, right? These are clearly for motility. And believe it or not, even though it's just a bacteria, that motility can be actually directed. So the bacteria actually can move, for example, toward food and away from like a poison and toxin in solution or on a petri dish, which is kind of amazing. And then last but not least, a special structure that some bacteria have, but not all, just like these other ones I've been drawing. One other structure called an endospore. And this is some bacteria can make this. Again, all of these are some. Not all bacteria have capsules, flagella, fimbriae. They all have a cell wall though. The endospore is a protective structure. So what happens is the bacteria living its life, and then if the environment becomes such that it's usually a temperature or water that causes stress to the bacteria, bacteria get stressed out. I have too many tests right now. It usually is low water or a dramatic change in temperature. And what happens is, remember inside bacteria is its DNA. So I'm going to draw that in a different color. And I'm simplifying that DNA. I'm just going to draw it like this, single chromosome, double-stranded. This is supposed to be double-stranded, double helix DNA in the chromosome. So as soon as the stress happens, some bacteria have the ability to form an endospore, which means this. Draw the cell a little bigger, cell membrane, cell wall. First thing they do is make a copy of their DNA. So make a copy of the DNA. They go through replication. And then they transcribe and translate some proteins. And I'm going to draw them as a different color here. Purple, maybe. They start making this, what's called a spore wall. And they start laying down this compound. There's a few ribosomes in there, just essential stuff gets put into, it's kind of like the, end of the world bunker that people would have in their basements, right? So it forms this spore, and when it's inside the cell like this, we call it an endospore. And then what happens is the vegetative cell, this cell dies, and we're just left with we call it a free spore, just left with this. And that free spore, the cool thing about that is it can exist, just be dormant for years, up to thousands of years. And then when conditions get better, mostly when there's more water available and the temperature either is warmer or cooler, depending on what the bacteria likes, this one spore will go back to forming one vegetative cell. So it's not reproduction because we're not making two cells at the end. We have one cell, makes an endospore, the cell dies and the spore remains, and then the spore can germinate and form one cell. So we talked a little bit about binary fission. Just gonna not talk too much more about that. You can skip the section on genetic recombination. I'm checking the tests first before I actually tell you that for sure. Yeah. In my book, which again is the 11th edition, not the 12th, in my book, it is section 27.2. So 27.2 is a skip. I'm not going to ask you questions about genetic recombination, transformation, transduction, conjugation. Come to microbiology, come to genetics, learn more about it. We just don't have time. Okay, but we do have time to talk a little bit about some of the nutritional modes of bacteria, which again helps reiterate some of the basic things we learned way back in the early chapters of the book. So bacteria are the most diverse as far as nutrient obtaining than any other organism on the planet. So we usually divide it up into, first and foremost, what do the organisms use for energy? And your choices for energy source are either going to be light energy from the sun or chemical energy. Now, what's different about this than what we talked about when we talked about the materials and glycolysis, cellular respiration? This chemical energy can be organic or inorganic chemicals. So that's a difference for bacteria than what we've talked about before. if the organism uses light, that's really what phototroph means. Notice I didn't say autotroph yet, right? Light energy is a phototroph. Chemical energy, we refer to those as chemotrophs. Right? Troph for food, chemicals for food, light, I guess, yes, for energy source. Now, we layer onto that what they use as their carbon source. Change colors here. Both of these, right? So I'm going to draw a line straight down and I'm going to put carbon source. And remember, why do we care about this? We care about this because we need carbon to build, right? Again, all the organic molecules that a cell needs. So again, think about building all the molecules in your favorite chapter, chapter 5. Same here. These organisms need a carbon source to be able to build proteins. I'll start simple: amino acids, DNA, RNA, nucleic acids, sugars, monosaccharides, polysaccharides, lipids. Now, depending on what they use for their carbon, that's going to help divide them into their last little groups here. So this, I know the green doesn't show up very well, so I'll try to use something else over here. So if you are an organism that uses light for your energy source, but for your carbon source, you use carbon dioxide, then you are what we call a photoautotroph. And if I asked you for an example of that, most of you would probably say a plant, right? But there are bacteria that are photosynthetic. So I will put plants, algae, and bacteria. Right? Now, if you use not carbon dioxide as your carbon source, but chemicals, so they're going to have to be organic chemicals, right? Something bigger than carbon dioxide, then we call you a photoheterotroph. Because you're not using light as that's where the photo comes from, right? Using light for your energy source, but you're using chemicals, something other than self, right? You're not being able to generate just from carbon dioxide for your carbon source. And believe it or not, there are lots of photoheterotrophs in the bacteria. They would be like the purple and some of the green sulfur bacteria. Over on this side, so we're using chemicals for our energy source. And again, believe it or not, there are some organisms that use chemicals for their energy source, but carbon dioxide for their carbon source. And they would be then called a chemoautotroph. Chemoautotroph, because they can make their own molecules from just carbon dioxide as their carbon source self, right? They're using chemicals for their energy source. And of course, there would be bacteria in this category. And then last but not least, if you use chemicals as your energy source and your carbon source is other organic molecules, right, or chemicals, then we would call you a chemoheterotroph, which would be you, right? Animals and most bacteria. And of course, fungus as well fall in that category. So the point of this is not only should you know these definitions, but the point of it is that bacteria are in all categories because they are the very bottom of the food web, right? They are the only kinds of cells, earliest forms of cells, as you can read about and talk about in chapter 25. And so first bacteria, and most likely first photoautotrophic bacteria. And from that, the rest of cells evolved over time, billions of years, but nonetheless. Then there's a little section about their oxygen requirements, because again, we're trying to introduce you to all the diversity in bacteria. And I would say this chart is in a table form in your book as well. So I'm going to erase and add. Oxygen requirements. And I'm just going to tell you about two. There are bacteria that we call aerobic, right? And or aerobes, S-A-B-E. And they, of course, would require oxygen for their metabolism, because they are going to engage in cellular respiration, aerobic respiration. And then there are organisms that are anaerobic, or anaerobes, and they live and reproduce and grow completely in the absence of oxygen. Right? So they're not going to do aerobic respiration. These are the organisms that are going to engage in what we, again, back to chapter 9, when we talked about aerobic respiration, we also mentioned anaerobic respiration. Right? I'm not putting fermentation on this list because most of the organisms that ferment exclusively can do it in the presence of oxygen. So this actually has other connotations, not just whether they use oxygen for their metabolism, but if they can get rid of the toxic forms of oxygen that are produced during metabolism. So we'll just leave it at that for now. Again, come to microbiology. We'll learn more about it. And the last thing that's really important that we should mention is about nitrogen fixing, because this is super important to basically life on Earth. Pretty much, there are only bacteria that can take the gas form of nitrogen, so like N2 gas, and convert it into things like nitrates and nitrites. And these nitrates and nitrites, these and ammonia, I should say ammonia too. These are usable forms of nitrogen for all other living things. I'm even going to make it simpler than that, all other living cells. So until organisms could fix nitrogen, because nitrogen is one of the most abundant gases in the air, but we can't use it. Humans can't use it either. We can breathe it in all we want, but we can't use it to make amino acids or to make nitrogenous bases, which make DNA and RNA. Think about uses for nitrogen, right? Predominantly, we need it for ATP to make the adenine, so that we can make more energy. So nitrogen is really, really, really important, and only a handful, only some soil bacteria and some water bacteria can actually convert the gaseous nitrogen in the atmosphere into usable forms for us and put it in the soil so that plants can take it up and fungus can take it up, and then we can eat those things, right? And just ingest those things, and then we get these usable forms of nitrogen that we can turn into nucleic acids, amino acids, the amine group that has to go on every amino acid to make proteins, because proteins do everything in cells. There's a nice picture in your book of some nitrogen fixers. And then, last but not least, there's a little bit to talk about for the archaea. I did want to mention the domain. And the three kingdoms, I think they've expanded to 5, but we're just going to talk about 3. The methanogens, the extreme halophiles, and the extreme thermophiles. So the methanogens can literally convert methane into usable forms of carbon dioxide and carbon monoxide. So they use that for their metabolism. Extreme halophiles, halo is for salts. These are extreme salt lovers. So they live in places like caves and Great Salt Lake, Dead Sea, places where there's really super high salt concentration. And the extreme thermophiles, heat loving, right? And I wanted to just highlight just super briefly, right? It's one of these extreme thermophiles that produces the enzyme that's called Taq polymerase. And that's the enzyme that can withstand the heat of PCR, which is, of course, what we're using for testing for COVID virus, right? So this is hugely important, right? These bacteria are found in hot springs, volcanic vents at the bottom of the sea, extreme environments. And then there were scientists that were able to cultivate them and then purify some enzymes out of them that we can use for biotechnology, right? So you never know where the source of some advantage is going to come from bacteria. All right, so I think I'm going to move back over here again. I made a list on your handout of bacteria in the environment and like sort of things that they do for us, advantages that we get from bacteria. And I was just going to put those on this whiteboard real fast. Just make a list for you. I know you can look it up, but so bottom of food web, we already mentioned that, right? So they're making all of our nitrogen for us, as well as contributing all kinds of basic organic molecules that then we don't have to make from scratch. Those bacteria, those cyanobacteria, you'll recognize them from, if you've already looked at, excuse me, chapter 25, those are probably some of the relatives of the first living cells. They produce oxygen as a byproduct because they're photosynthetic, right? We need oxygen for aerobic respiration. And, 4 billion years ago, there wasn't any oxygen until there were enough bacteria to produce the oxygen for us. I lost my box. Okay. Nitrogen fixers, I already mentioned those, but put that on the list. Bacteria are the major decomposers with the fungus, of course, breaking down everything that we make. There's scientists trying to particularly engineer bacteria that can digest plastic faster than normal so that we can help clear the landfills and clear the plastic waste in the oceans. Food production for humans and animals, right? So I would list things there like alcohol. I know that's not really food, but alcohol, vitamins. We use the bacteria they produce the vitamins. We genetically engineer them so they produce extra, and then we purify the vitamins out of the bacteria. Obviously, cheese, yogurt, anything that's fermented, right? Like that fizzy drink we're drinking for our health, right? Kombucha, right? That, as well as like sauerkraut and pickles and any other kind of fermented food. Put crop technology on here. We're using bacteria to make crops such as corn and beans resistant to infections with particular insects. The insects ingest the bacteria makes a toxin, kills the insects, but doesn't affect humans or the plant growing. And then bioremediation, which I already mentioned. I don't think that's spelled right. Looks like we can use bacteria to actually make biodegradable plastics. And then I also put on the list bioremediation. I guess biodegradable plastics are not really a food, so let's give them a little, put them out there with the rest of these. Bioremediation means using bacteria basically to clean or clear compounds from the environment, such as, right, so the biggest example of bioremediation is sewage treatment. We use bacteria to digest most of the sewage that then just becomes a sludge of solid waste. And then there's other bacteria that can be used to degrade that even further. And then with oil spills, there are bacteria that you can use that you have to give them an extra source of nitrogen and phosphorus, but you can inject that into the water or put that in the water. The bacteria grow, they digest and literally decompose the petroleum and the petroleum distillates, and then gets into smaller and smaller and smaller globules that eventually lessens the impact on the environment. I think there's some pictures of some people doing that. And I would also put on this list fuel production, right? So there are people trying to use bacteria, particularly E. coli, I think, to make biofuels instead of relying on petroleum distillates for driving our cars. All right, and then I did list crude oil spills, research and technology, so research, so biotech, and relationships. That's not important. I'll leave that one off. All right, so that's kind of that list and a little bit more about some of the things on that list. And the last thing, usually I ask students if they have any bacterial infections that they would like to know more about, since I teach microbiology. And then I usually go on about talking about different bacterial infections. Your textbook lists Lyme disease and looks like E. coli O157H7, and kind of think, see what else they do. They do a short thing about C. diff and salmonella. But I was curious if anybody, just go ahead and shout out or put it in the chat. Anybody have a particular bacterial infection that they would like to know more about, and then I can talk about it, and then I would put that one, a question about it on the exam. Yes, no, maybe. If not, then I'll just use the ones that are in your book. So it's up to you. I think in the summer, it looks like I have some scribbled notes on my paper about Clostridium, tetanus, botulism difficile, and listeria. I don't know why I did listeria, but all right, then if not, like I said, I'll use the ones in my book, or I will use those. So that's good. And anything else you have questions about content material? Yes, no. All right, I'm going to end the recording then, and